Hi, this is Pierce Paul, corn and small grains pathologist and extension state specialist in the Department of Plant Pathology at The Ohio State University. In today's presentation, I'll focus on the management of Gibberella, ear rat, and vomitoxin in corn with fungicides. However, since there's very little research done on fungicide efficacy against Gibberella, ear rat, and vomitoxin in corn, I'll use lessons learned from research with Fusarium head blight, a related disease that's been more widely studied, to direct my presentation on how to use fungicides to manage Gibberella, ear rat, and vomitoxin in corn. Specifically, I'll talk about similarities between the two diseases, similarities between the infection process, and what implications those similarities have for managing these diseases with fungicide, again emphasizing Gibberella, ear rat, and vomitoxin in corn towards the end of my presentation. Our production system is one that favors the development of Fusarium graminearum causing diseases. It infects corn, wheat, and soybeans. It also survives in crop residue in the field. So since most of our fields are either on the no-till or some form of reduced tillage, the fungus can survive from one growing season to the next. And once conditions become favorable in the spring, then it can infect the crops, causing Gibberella, ear rat in corn and Fusarium graminearum in wheat. Spores are produced in crop residue. They are splashed or wind disseminated to wheat spikes and to corn ears, where they infect during the pollination growth stage, in the case of wheat during anthesis, and in the case of corn during silking, that R1 growth stage. So if you look at this disease cycle and how the disease develops, it's clear that some form of crop residue management, such as tillage, can help to reduce the severity of this disease. Also, some form of crop rotation. If you rotate away from the crops that are most susceptible to the disease, it can help to reduce the severity of disease. There are many similarities between Fusarium head blight and Gibberella, ear rat, other than the fact that they're caused by the same fungus, Fusarium graminearum, also called Gibberella zeae. One very important similarity that has direct implications for management with fungicide is the fact that in both cases, the fungus infects during pollination. In the case of wheat, the fungus enters or infects the spikes during anthesis, and in the case of corn, the fungus infects during silking. Spores land on those silks, germinate, penetrating the ears, colonizing the developing grain, and produce mycotoxins. Once conditions are wet and humid, this process occurs much quicker, and as a result, fungicides are needed to protect. So in both wheat and corn, fungicide applications for control of Gibberella, ear rat, and Fusarium head blight should be made during this pollination window. Years of research have been conducted looking at different timing of fungicide to control Fusarium head blight, less so in corn. And we've seen over these years that the best results are seen when the fungicides are applied during the pollination window. Fungicide can provide protection when applied early, applied late, as you'll see in a little bit, but the best results are seen when those fungicides are applied during pollination, during anthesis, in the case of wheat. And we've done something similar based on what we've learned from our work with Fusarium head blight. We've developed a few research projects looking at fungicide timing in my lab, and other researchers have done the same as well. And we've found that some of the best results are seen when the fungicides are applied during silking, that pollination window in corn. In terms of the impacts of Gibberella, ear rat, and Fusarium head blight, both lead to grain yield and test weight reduction, contamination of grain with mycotoxins, and consequently, reduction in the market value and utilization of the crops. Grain heavily contaminated with mycotoxin may receive a lower price at the point of sale or even rejected. In addition, a big concern is feeding contaminated grain to livestock. Monogastric animals are particularly sensitive. Swine, if consumed high levels of vomitoxin contaminated grain or feed, can lead to vomiting and feed refusal, which leads to lower rate of conversion. There is no single strategy that can be used to reduce the level of vomitoxin contamination of grain. However, multiple strategies can be integrated to reduce toxin contamination, as well as reduce the level of disease in the field. In terms of mitigation strategies, once grain is harvested from fields affected by Fusarium head blight or Gibberella A rot, nothing can be done in terms of fungicide application to reduce the level of contamination in storage. However, cleaning and harvesting strategies can be used to minimize the overall level of contamination. Since both diseases cause shriveled lightweight kernels, as well as contamination of grain with fines, cleaning strategies can help to remove those lightweight kernels and help to remove fines from the grain lot, can also lead to a reduction in the level of vomitoxin contamination. Fines are often more heavily contaminated with vomitoxin than the grain itself. Similarly, harvesting strategies can be used to remove shriveled lightweight kernels as well as remove fines during the harvest process, and this again will help to reduce the overall level of grain contamination with mycotoxins. In the fields, several strategies can be used. The first would be to select the best resistant variety or the best resistant hybrid you can find. Hybrids vary in their level of susceptibility to Gibberella A rot. And if you find a hybrid that's resistant to the disease, this is the first and most important step to help reduce the overall level of grain contamination and the overall level of disease severity. Tillage and crop rotation can also help to reduce the level of disease severity and grain contamination with mycotoxins. Since the pathogen survives in crop residue in the field, tillage can help to reduce the overall survival and the overall level of infection and disease development. Crop rotation away from a host crop can also help to reduce the level of disease and grain contamination with mycotoxins. However, since this pathogen can be blown easily from one field to the next, even fields rotated away from a host crop or fields planted with minimum in-field crop residue can still have problems with Gibberella A rot and Fusarium head blight. I'll focus most of my presentation on fungicide application with an emphasis on products, application timing, and a little bit on application technology. One of the fungicides I'll focus on during my presentation is called Miravis ACE. This is a new fungicide that was recently labeled for managing diseases of small grain crops. For years, we've focused our attention on the use of triazole fungicides alone for the control of Fusarium head blight. But now this fungicide gives us a new option in terms of using a fungicide made up of two active ingredients, a triazole, or also called DMI, and an SDHI. This is good news in terms of fungicide resistance management. Using fungicide with mixed chemistries can help to reduce the development of fungicide resistance. So what I'll do, I'll share some results in terms of the efficacy of this new fungicide compared to the efficacy of Prosaro and Caramba. Those are considered the industry standards for scab management, as well as look at timing of Miravis ACE in terms of the control and reduction of vomitoxin and Fusarium head blight in wheat. For years, we've recommended that fungicides be applied strictly during anthesis to effectively control Fusarium head blight and vomitoxin in wheat. However, this new fungicide, Miravis ACE, has on its label that it can be applied as early as fixed growth stage 10.3. That's when 50% of the head is seen emerging from the leaf sheath of the flag leaves. So if you walk fields and you see tillers with 50% of the head emerging from the leaf sheath of the flag leaf, that's the growth stage at which the label says that this fungicide, Miravis ACE, can provide adequate protection against Fusarium head blight and vomitoxin in wheat. We also recommend fungicide application, or we also seen or tested fungicide application at the full head emergence growth stage. So what I'll do is share some results looking at these early timings relative to the anthesis timing, and I'll also look at some late timings to see how effective the fungicide works in terms of protecting against Fusarium head blight and vomitoxin in wheat. And by extension, we did some similar work looking at fungicide timing for efficacy against Gibberella A rot and vomitoxin in corn. Since the fungus infects the spike during anthesis, it's important to protect the wheat heads during this critical growth stage. However, anthesis is not only a single point in time. We've got early anthesis, when anthers are extruded in the center of the spikes. We've got mid-anthesis, when anthers are extruded towards the tip of the spike, and late anthesis, when anthers are extruded towards the base of the spike. This could be a window of about 5 to 7 days, depending on the weather conditions. An application of fungicides during this window is what we found to provide the best results in terms of reduction of Fusarium head blight and vomitoxin in wheat. Here we have side-by-side comparisons of Prosaro, Caramba, and Miravis Ace when applied at that Anthesis or fixed 10.5.1 growth stage. On the far left, you've got the untreated check, no fungicide was applied, and clearly you can see plots treated with Prosaro, Caramba, or Miravis Ace, they've got visually less disease than the untreated check, providing some evidence that these fungicides may be similar in terms of their efficacy against visual symptom development. Here we've got a side-by-side comparison of fungicide programs in terms of their efficacy against visual symptoms. Visual symptoms is measured here as Fusarium head blight index, which is a percentage of the spike area covered with symptoms of the disease. In the check, on the far left in the red bar, you've got about 15% Fusarium head blight index in the check. All fungicide treatments resulted in significant reduction in visual symptoms in terms of index relative to the untreated check. Application made at anthesis, early anthesis, or late anthesis, or even early heading, that green bar Miravis Ace applied at early heading, reduced disease severity by about 56 to 68%. Double application, application of Miravis Ace followed by Prosaro, or application of Miravis Ace followed by Caramba, application of Miravis Ace followed by Fungicide, reduced disease severity by about 77 to 87%. So two treatment program can provide even greater reduction in the overall level of visual symptoms. Miravis Ace was applied at anthesis, early anthesis, and then Prosaro, Caramba, or Fungicide were applied 4 to 6 days after early anthesis. In terms of vomitoxin, this graph similarly shows side-by-side comparisons of different fungicide programs for vomitoxin reduction. Again, vomitoxin in the far left in the red is about 5 parts per million vomitoxin in the grain. And as you can see, all fungicide programs resulted in significant reduction in vomitoxin levels in the grain. What I want you to note here, anthesis fungicide programs reduced vomitoxin levels, that's early anthesis, between 33 and 44%. Application towards the end of that anthesis reduced vomitoxin by 59%, close to 50%. But application early, in terms of applying Miravis Ace at that early heading growth stage, only reduced vomitoxin by about 12%. So basically what this is showing, a single application when made at anthesis, early anthesis, or late anthesis, provide greater reduction in vomitoxin levels in the grain than a single application at that early heading growth stage. Again, as was the case with visual symptoms, a two-treatment program applying Miravis Ace at anthesis, followed by Prosaro, Caramba, or Fungicide 4 to 6 days later, resulted in about 61 to 71% reduction. In other words, the two-treatment program resulted in the greatest level of reduction in vomitoxin in the grain. While Fusarium head blight and vomitoxin were the focus of these fungicide programs, Fusarium head blight and vomitoxin are not the only problems that affect wheat in terms of diseases. So what we did is collected data on the efficacy of these programs against leaf diseases, specifically Septoria and Stag and Ospor leaf blotches. If you look at total leaf blot severity in the untreated check, this is leaf blot severity on the flag leaf. It was about 25% in the untreated plots, and all fungicide programs reduce the level of disease severity close to or below 10%. In other words, all fungicide program, anthesis application, early heading application, two-treatment program, and even late anthesis application all reduce the level of visual symptoms in terms of leaf diseases caused by Septoria and Stag and Ospor. In a year such as 2020, with fairly high levels of Fusarium head blight and leaf diseases, all of the tested fungicide programs resulted in significantly higher grain yield than the untreated check. However, the magnitude of the yield response, in other words, how much yield increase occurred, depending on the fungicide program. In green bars, we've got some of the highest yield responses in general. Miravis Ace, followed by Prosaro, Caramba, or Fungicide, resulted in highest yield response. But across the board, all fungicide programs were effective enough against leaf diseases as well as head scab to result in a significant yield increase compared to the untreated check. So what about the efficacy of fungicide against Gibberella, ear-rot, and vomitoxin in corn? We observed from our research with Fusarium head blight that when applied during that pollination or during that anthesis window, fungicides were most effective against Fusarium head blight and vomitoxin. For Miravis ACE, that new fungicide, when application was made at early heading, that's well before the pollination period or well before anthesis, the efficacy against vomitoxin was much lower than when that same fungicide was applied during anthesis. So by extension, you would think that fungicides would be most effective against Gibberella, ear-rot, and vomitoxin in corn when applied during that pollination period, that is, during the silking window. We'll take a look at some results from my own trials and results conducted by other researchers on the efficacy of fungicide against Gibberella, ear-rot, and vomitoxin in corn. Again, I'll present some results on the efficacy of a new fungicide called Miravis Neo. This is a sister fungicide to Miravis ACE that's used in wheat and other small grain crops. Miravis Neo is labeled for use in corn, and like Miravis ACE, it's a mixture of multiple active ingredients. In this case, it's a mixture of a triazole, a strobilurin, and an SDHI. This is a combination premix of multiple active ingredients. And I'll share some results from some trials we conducted looking at the efficacy of this new fungicide against Gibberella, ear-rot, and vomitoxin in corn. As was the case with Fusarium head blight, where fungicides were most effective when applied during that window of susceptibility to infection, the same is true here in corn. Remember, the fungus infects via the silks, so fungicides need to be applied to protect those silks from infection. So what I'll share here is some results where fungicides were applied at silking, before silking, and after silking, and we'll compare efficacy in terms of reduction in Gibberella, ear-rot severity, as well as vomitoxin contamination of grain. Far less work has been done on the efficacy of fungicides against Gibberella, ear-rot, and vomitoxin in corn. So what I did in order to get some data for my presentation, I searched the literature, and here I'm going to summarize results from work done by my colleagues. Here are some data from studies conducted at Purdue, where they looked at 3 different fungicides. Headline, called Pyra, Proline, labeled here as Prot, and Quilt XL, labeled here as ACE plus Pro. Of these 3 fungicides, only Proline resulted in a significant reduction in the level of visual symptoms compared to the check. Here, visual symptoms is measured in terms of disease severity, which is percentage of the ear covered with symptoms of the disease. The check had about 22% of the ear covered with disease, whereas the Proline treated plots had about 10% on average of the ear covered with the disease. So the only fungicide here in this trial that resulted in a significant reduction of visual symptoms was the Proline treatment. However, in terms of reduction in vomitoxin in the grain, even though there was a numerical reduction with Proline and Quilt XL, reducing the level of vomitoxin in the grain by about 2 to 3 parts per million, this difference was not statistically significant, suggesting that in this particular trial, with the level of disease observed in this field, or the level of vomitoxin observed in the grain, these fungicides were not effective at reducing vomitoxin. In these trials, ears were artificially inoculated. In other words, the fungus was injected directly into the ears, and this resulted in fairly high levels of disease and vomitoxin. These levels are probably higher than levels you would see in the field. So it would be good to reevaluate these fungicides under natural infection conditions to see if they actually reduce Gibberella, ear-rot, and vomitoxin. In a second trial, again at Purdue, all 3 fungicide programs resulted in significant reduction in the level of visual symptoms in terms of disease severity on the ears. In this trial, levels of disease was much higher. Close to 60% of the ear was affected by the disease, whereas in fungicide-treated plot, the level of disease severity was reduced to about 40%. In other words, a 20% reduction in the level of disease compared to the untreated check. However, again, even though these 3 fungicides reduce the level of visual symptoms, when you look at the efficacy against vomitoxin in the grain, they were not significantly different from the untreated check. The untreated check had about 32 parts per million vomitoxin. There was a numerical reduction of vomitoxin level with some of these treatments, but these differences were not statistically significant. This is because fungicide efficacy against Gibberella ear rot and vomitoxin in corn is known to be highly variable. And when you've got high variability among reps of the same treatment, then it's hard to separate treatment in terms of efficacy against vomitoxin. As was the case in the previous trial conducted at Purdue, these ears were artificially inoculated, which resulted in fairly high levels of disease and vomitoxin. These levels were probably higher than you would see in the field. So again, it would be useful to revisit and reevaluate these fungicides under conditions that are comparable to what you would see in the field, where the overall level of vomitoxin levels might be lower than you would see in an artificially inoculated trial. Here are some more results from trials conducted in Santa Catarina, Brazil. Here again, like the study in Purdue, they inoculated ears with the fungus and then evaluate the efficacy of a fungicide treatment consisting of 3 active ingredients when applied at different times relative to early silking, in other words, R1. They looked at the efficacy of this fungicide when applied within the first 2 days after silking, before or after silking, within the first 4 days of silking, and within the first 6 days of silking. And they compared the efficacy on 2 different hybrids. In both cases, the greatest reduction in the level of disease, the lower bars here, was observed when that fungicide was applied within 2 days of silking. As the fungicide applications were made earlier or later relative to that silking window, efficacy decreases, with applications made 6 days after silking having levels of disease very comparable to the level of disease observed in the untreated check. Unfortunately, this trial does not report results on vomitoxin in the grain. But it serves to show that, again, fungicides are most effective in terms of reducing visual symptoms of Gibberella ear rot when that fungicide application is made close to that silking window, in this case, within 2 days of silking. As fungicide applications are delayed or made earlier, the efficacy is reduced and the results tend to be more variable than when that fungicide application is made close to that silking window. In another trial conducted looking at fungicide timing for Gibberella ear rot and vomitoxin control, here this trial was conducted in Canada, and they looked at Proline applied at silking, before silking, and after silking. And as was the case in the trial conducted in Brazil, this time they're showing vomitoxin results, as well as results for another toxin that's produced by Fusarium graminearum called zeae. That's the purple bar. The green bar shows vomitoxin results. The greatest reduction in vomitoxin compared to the check was observed when that fungicide was applied at full silking. Application made at V18, tasseling, 5 days after silking, 10 days after silking, resulted in lower levels of reduction in vomitoxin compared to the check. Again, emphasizing the importance of getting that fungicide on to protect those silks when the crop is most susceptible to infection. This resulted in the greatest reduction of vomitoxin as well as zeae, the other toxin produced by Fusarium graminearum. It is important to note that in a trial conducted in Canada, where they saw the greatest reduction in vomitoxin when that fungicide was applied at full silking, they used drop nozzles. In other words, instead of applying that fungicide using a high clearance sprayer where the fungicides are delivered close to the height of the tassels, they used drop nozzles where the nozzles, the nozzles were extended into the canopy, and that fungicide was applied close to the height of the ears. This resulted in the greatest reduction in vomitoxin in that particular trial. This suggests that fungicide application method may be important in terms of reducing vomitoxin in the grain. Here in Ohio, we conducted trials to look at the efficacy of fungicides when applied using drop nozzles. Again, we inoculated ears with the fungus and then applied these fungicides at the R1 or the silking growth stage. We looked at Prosaro, Headline, Headline Amp, Miravis Ace, and Miravis Neo. All fungicide-treated plots had lower levels of disease than the untreated check, but the greatest reduction in disease severity was observed when Headline AMP as well as Miravis Neo were applied. Headline provided the lowest level of reduction. Even though the reduction was numerically different from the check, it was considerably lower than plots treated with Headline AMP and Miravis Neo. When we look at vomitoxin levels in the grain, again, Proline, Headline AMP, and Miravis Neo resulted in the lowest levels of vomitoxin in the grain. In this case, the grain had about 25 parts per million vomitoxin, and Miravis Neo reduced vomitoxin levels to about 10 parts per million. Of note here, Headline did not reduce vomitoxin relative to the check. In fact, the Headline treated plot had slightly higher levels of vomitoxin than the untreated check. Here are some more results from a second trial, this time with much lower levels of disease in terms of the efficacy of the same 5 fungicides against Gibberella, Erat, and Vomitoxin. The first trial with the higher levels of disease was conducted at South Charleston. This trial was conducted in Worcester. Again, all plots were artificially inoculated. The untreated check in this case had less than 0.2% of the ear covered with visual symptoms. And in this case, all fungicide treatments resulted in lower levels of disease than the untreated check. But again, the overall level of disease was generally low to start with. In terms of vomitoxin, the untreated check had about 2 parts per million vomitoxin, and all fungicide programs reduced vomitoxin levels to about 0.5 or lower parts per million vomitoxin. What are these results suggesting? That drop nozzles can be useful for controlling Gibberella, Erat, and vomitoxin in corn. The efficacy depends on the fungicide, and it depends on the overall level of disease. When disease levels are high, we saw better results with Miravis Neo, Headline AMP, and Miravis ACE than we saw, as well as Prosaro, than we saw with Headline, especially in terms of vomitoxin reduction. However, when the overall level of disease was low and the overall level of toxin was low, all 5 fungicide programs resulted in lower levels than the untreated check. These results are only from a couple of trials conducted during a single growing season. We did repeat some of these trials in 2019, but the level of disease and vomitoxin levels were way too low to be able to compare treatments. We plan to repeat some of these studies, again, to evaluate how consistent these results are in terms of efficacy. So in summary, fungicides for Gibberella, Erat, and vomitoxin control. We saw promising results with multiple fungicides, even though the efficacy tends to vary considerably. It depends on the fungicide and depends on the overall level of disease and toxin in the trials. More research is needed evaluating other fungicides. We want to revisit application technology in terms of how these fungicides are applied, high clearance versus drop nozzles. Drop nozzles seem to provide the best results, but again, more research is needed to say more conclusively how effective fungicides are against Gibberella, Erat, and vomitoxin in corn. For the Fusarium head blight research, the data I presented were from trials conducted across the country by several colleagues. So I want to acknowledge my colleagues, as well as the U.S. Wheat and Barley Scab Initiative, providing funding for this research. And then for the Gibberella, Erat research, some of the work done here on fungicide efficacy was supported by the Ohio Corn and Wheat Growers Association, and I want to extend my gratitude to them, as well as to Bayer Crop Science and the USDA NIFA for providing funding for my Gibberella, Erat research. That's all I've got. Thanks for listening.